Now today's video is on embryology definitions, and embryology is basically the study of development of the embryo from the stage of fertilisation, so from when the sperm first fertilises the egg until the fetal stage. And that's not meant to be, well, it's a fetus but it doesn't look like one, but anyway. So some key definitions. In week 1 we have cleavage. And now cleavage is basically mitotic divisions of the fertilised oocyte, so the fertilised egg, and it forms a blastula from a fertilised ovum. So here's the fertilised egg, and it's going to go through mitotic divisions, and it's going to keep dividing and keep dividing, and it's going to form a blastula. And however, it's important to remember that through cleavage, the size stays the same. So this fertilised oocyte, even though it's continuously divided by mitosis, it's not going to get any bigger, because it still needs to fit through the fallopian tubes and it still needs to make its way to embed in the uterus. And so even though all these divisions are happening, it stays exactly the same size. And this means that it can fit through the isthmus. I don't really know how to say that, but the isthmus. And this here is basically just the narrowest part of the fallopian tube. So another word that's important in embryology is zona pellucida. And this is basically, it's a tough glycoprotein coat that surrounds the oocyte. So it surrounds, here's our fertilised egg. Yeah, it's all happy because it's got a sperm and it's ready to embed in the uterus. But the zona pellucida, because this egg, this fertilised egg, doesn't want to implant too soon. Because if it implants too soon, then it's not going to be able to implant in the uterus and it's going to implant somewhere else. And that's called an ectopic pregnancy. And that's just where it, well, for example, it can implant in the fallopian tube or on the external wall of the uterus or in the gastrointestinal system. So basically the zona pellucida is this tough glycoprotein coat that's surrounding the oocyte, surrounding the fertilised egg, and that's just to stop it implanting prematurely. Okay, and now we've also got the morula. Now morula is Latin for mulberry, and that's because a morula basically kind of looks like a mulberry, so it's like loads of cells that are all closely clumped together. There we go. And a morula is a solid ball of cells, and this results from the division of a fertilised ovum. So here's our little fertilised oocyte, and here's the zona pellucida, and mitotic divisions of this forms the morula. And the morula is very important because the morula is going to form our blastula. And this is all happening in week 1 of embryology, but I'll go through this in more detail in a different video. But basically the morula is going to become the blastula, and the morula starts to form, I think it's 4 days after fertilisation. So 4 days after fertilisation you get your morula, and then this is going to become your blastula. Now the blastula, it's a hollow ball of cells. So unlike the morula, which is a solid ball of cells, the blastula, it's a hollow ball of cells. And our blastula is very important because this is what's going to become our embryo. And the blastula, these cells here, they surround, this is a fluid-filled cavity. And basically a blastula is formed by cleavage. So a blastula is formed by repeated cleavage of our fertilised egg. Here's our fertilised egg, all happy. It's going to divide to become the morula. The morula is then going to form the blastula. And the blastula, we have an inner cell mass, which I'm going to label ICM here. We've got the inner cell mass, and we've got the trophoblast. Now, I'll talk about these more in a different video, but the inner cell mass is basically going to go on to become the embryo and the extra embryonic membranes, and the trophoblast, I think, is going to become the placenta. But I'll double check that and we can have another video on the details of the development, but this is mainly just on definitions. So the thing you need to remember about the blastula is it forms in week 1, forms from the morula, and it's composed of an inner cell mass, trophoblast, and this fluid-filled cavity here. So this is just the fluid-filled cavity. So that's all in week 1. Now, in week 3, we're going to skip ahead to week 3, you get gastrulation. And gastrulation is the formation of the 3 germ layers, the ectoderm, the endoderm, and the mesoderm, and this happens in week 3. And this is a result, these 3 layers, these 3 germ layers that form here, this is called gastrulation, the formation of these layers, but this is a result of cell division and migration. And ectoderm is the outer layer, endoderm is the bottom layer. I'm going to put A for outer, B for bottom, and mesoderm, just remember this M for middle, M for mesoderm, is in the middle. Now, another word that's important in embryology is potency, the potency of cells. And this just basically describes a cell's ability to differentiate into other cell types. So for example, we've got totipotent, unipotent, and multipotent. Now these are just a few examples, and I'll go through them in more detail in a future video.